Yeah, this will be a bit of a change in pace, sort of a shift from some of the neurophysiological talks that we've been seeing so far, which have, of course, been fantastic, into more of behavior. And as Reza mentioned, I am presenting from Seattle. I'm here for the holidays at the house of my family. I'm literally on the living room couch right now, so I apologize for any potential background noise. Um, my talk will be titled Effort Cost of Reward Affects Decisions and Movements of Marmosets During a Foraging Task. And notably, a lot of this work and inquiry really came about quite serendipitously. We never really planned for the behavior that we saw, but kind of took a deep dive into it anyway because it was fascinating. Much of these analyses and, of course, my general thinking of the results is still ongoing. So I would, of course, love any feedback, criticisms, potential ideas. And I can't see anyone, so by all means, please just interrupt me if you have any questions or need clarity on anything. And with that, I'll get started. So here's a bit of an outline into the talk. We're going to start with some background where we'll use a crow as our teacher to demonstrate some decisions that are made during foraging. We'll then take a look at the effects of reward and effort, foraging variables on how movements are affected rather than just decisions themselves. And we'll take a look into how subjective value can often be inferred from vigor of movements, which is, in my opinion, really fascinating. From there, I'll describe some of the methods that we use in this study. Specifically, I'll give you a description of marmosets as a non-human primate model, and frankly, why they're a fascinating foraging primate model as well. And I'll describe to you the foraging task and the structure of the experiment that we engaged them in. We'll then dive into some results, namely the effect of effort on foraging decisions, the effect of both effort and reward on movement vigor during foraging. And then I'll present to you guys some patterns in behavior that might prove some evidence into a link between sort of internal states in the brain and movement vigor. And then finally, I'll try and sum everything up together and hit you with a sort of so what, why might this be important and what implications might this have on our understanding of underlying neural substrates of the behavioral phenomena that we're seeing. So I'm going to start this talk in a nature reserve in the village of Dorset in the southwestern part of the United Kingdom. It's quite a beautiful place, actually. And the reason we're going there is because this generous YouTuber posted this fascinating video of a crow foraging for clams on a beach, actually. And what you'll notice in this video is that, if I can get this video to work, just a quick moment, is that the crow begins by looking for clams and then eventually digging them up. And they leverage gravity using this fascinating technique to break the clams open to get into the meat. And sometimes that's unsuccessful, and the crow has to do this repeatedly, as you see here. And eventually they're successful, able to crack open the shells and get to the insides, harvesting the meat. Now you can imagine that this is a pretty tough way to make a living, if you will. And I can't help but question, is there some optimal strategy these crows take to go ahead and get their food in this manner? Let's just watch that one more time. The crow looks for clams, digs them up. When it's selected a clam, it flies into the air, drops it in an attempt to crack open the shell. Sometimes this is unsuccessful, so they have to do it again, repeatedly, until they're eventually successful. A lesson of life, if you will. So indeed, to survive in nature, it's crucial to make decisions that optimize for net energy expenditure during foraging, right? Because if you expend more calories than you ingest, you won't survive. And now, sticking with this example of crows, Richardson and Verbeek in '86 published a really fascinating paper where they observed crows doing this. And here's a figure from that paper where the y-axis is percentage of clams that the crows ended up foraging from, and the x-axis is clam size. And what you see is that these data cluster quite distinctly. Crows ended up, despite expending the initial effort to find and dig up the clams, abandoning small clams and harvesting from large clams. Now, this seems rather intuitive because, of course, if the reward within the clam is not justifiable enough to go ahead and fly into the air and drop it and crack it, then they should abandon it. Perhaps it's optimal to stick to large clams. Behavioral ecologists have developed a model called optimal foraging theory, which attempts to help us understand this decision-making process And in a fascinating sort of vast literature, it's been used to understand decisions made across many species. So worms, birds, monkeys, and even us as humans. And it makes one sort of major prediction, and that's that, and this is the first prediction I'll submit to you guys as a sort of motivation to the results I'll show you later. It's that harvest should only commence when the reward is large enough to justify the acquisition effort. Now, it seems quite intuitive. Now, these variables, these foraging variables of reward and effort, they actually affect more than just decision making. They influence the vigor with which we move. Here's an example in the saccadic eye movement system, which I find to be a fascinating system to try and get at some of these patterns, because of course you can't quite explicitly control the speed / vigor of your saccades. So here's some work from Esson in our lab, and it drives home the first sort of underlying point here, which is that we tend to move faster towards stimuli that promise greater rewards. So here what you see are saccades to stimuli being presented to the same eccentricity of your visual field. And what we notice is that humans tend to make saccades faster to faces than to noise, because of course we tend to value faces more, and faces are more valuable and, reward intensive for us to look at. The second point is that effort, the effort variable, tends to make us move slower. So we move slower towards stimuli that require greater effort. Here's some work from Terim in the lab, who presented two of the same stimuli at different eccentricities. We saw that in humans, we tended to make saccades faster to less effortful stimuli and slower to more grateful, and slower to greater effort stimuli. And this begs the question, right? So what is the justification of these vigor patterns? Why is it that the brain is allowing us to control our movements in these ways? Well, it's because of the fact that movements expend time and energy, and this in the end will discount the value of the promised reward. And this has a pretty tremendous implication, if you ask me, and that's that the brain tends to control our decisions and movements based on a common principle, which is to maximize utility. And in this case, we describe utility mathematically as the total sum of reward minus the total sum of effort in time. So we would like to maximize reward while minimizing effort. And this leads us to a second prediction here, and that's that if we increase the effort required to harvest from a food cache, the brain shouldn't only force us to work longer to increase the reward available, but rather to also move slower, to move with a reduced velocity. Now, understanding this link between utility and movement vigor has powerful implications, because from it, you can sort of infer the subjective value of a choice, the way in which we value a particular stimulus that we're deciding to move to. And a vast amount of work over the last decade or so has looked into this and has noted that during decision making, as humans and other animals deliberate, our movements reflect not just choice, but rather subjective value of that choice. And the typical thing that is seen in the results is that we tend to move with greater vigor towards higher value stimuli. And this is an amazing thing because it serves as a measurable behavioral proxy for a variable that we can't quite see. It's a hidden variable in the brain. So consider this: when we're presented with two different decisions, we tend to value our options differently. And reflected in that choice of options is also differences in the way in which we move to those options, and that also is influenced by how much we value them. However, there's a confound here. There's an elephant in the room, and that's that our internal state will oftentimes drive how we value things. On a hot summer's day, I might value ice cream more than soup, and that might flip when it's cold outside. And so here's a question for you. What can our movements tell us about our internal state, perhaps how we feel? Now, that also is a hidden variable, and it can be sometimes difficult to get at and measure behaviorally. However, we do have a window into this, a fascinating one, and that's the pupils. And we'll be looking at marmoset pupil size changes in this study. Pupil dilation is typically used as a proxy for activation of brainstem neuromodulatory system, and is used as a measure of all sorts of arousal, all sorts of states of the brain. It's been linked to a large degree of experimental variables. A few examples of these are stimulus salience and value, attentive state, changes in task utility, and it's even been linked to some fascinating behavioral phenomena. For instance, Wang et al. Demonstrated that the pupil size tends to predict saccade reaction times as well. So I have a third prediction for you, and the final one here. And that's that perhaps we can see a link between changes in pupil size and changes in movement vigor across multiple movement systems. So just to remind you, the first prediction is that a harvest should commence only when the reward justifies the acquisition effort, a decision-making phenomena, if you will. The second prediction is that if we were to further increase the effort required to harvest a food cache, the brain should not only respond by working for longer to increase the reward available at the time, but also to move with a reduced velocity to optimize for energy expenditure. And finally, maybe we can see a link between changes in pupil size reflecting a sort of change in the state of the brain and the way in which we move, changes in movement vigor. We'll be testing these predictions in marmosets today. So just to let you know a little bit about marmosets and why I think they're fascinating. Here's some background. They're small New World monkey. They originate in Brazil. They have an amazing array of different behaviors. They're social and they live in family units. They're vocal and they rely on species-specific sound production for their communication. And they're visual and they make saccadic eye movements to explore their environment. And notably for us, they're extremely efficient foragers and they rely on fine control of their tongue for their survival in the wild. The reason for this is because of how they feed. They rely primarily on plant exudates like gum, sap, and tree resin. And to get at this, they climb up trees, they gnaw holes in trees, and they use their tongues to extract the desired exudates. They forage essentially for this stuff. So here's the task structure. Basically, we make marmosets forage by working by making saccades to accumulate food. And then they harvest the food through licking. And we don't constrain them in their decisions at all. We let them work for as long as they want and then harvest for as long as they want. And we observe how these dynamics change in time during a task. So animals are chaired and head fixed, positioned in front of a TV screen. We're measuring their eye movements at 1,000 hertz and their pupil sizes at 1,000 hertz. And we're recording licking at 100 hertz. These animals are trained to follow targets on a screen by making saccades. And this is what this task structure looks like. They make a centripetal saccade from wherever their gaze is at a given point in time to the center of the screen and fixate. They're then presented with a primary target in one of 8 pseudo-random directions to which they make a primary saccade to. When we detect the primary saccade, we jump at target, eliciting a secondary corrective saccade. At the end of this sequence of saccades, they're given a tone and dispensed a small amount of reward. Now, I must note that this reward is actually the food that they consume, and we give them their entire day's worth of nutrition while they're doing the task. These animals are not water restricted, and reward is not juice. It's their actual food that they consume mixed with some applesauce. They really like applesauce. And also, as a secondary note, while these animals are performing this task, we're also recording from various regions of their cerebellum for other projects. So the task typically breaks down into 2 phases, 2 periods. There's a work period in which the animal is making the sequence of 3 saccades to accumulate food. We dispense them again, as I mentioned, a small amount of food per successful trial. And then a harvest period in which they elect to stop working and initiate a harvest. And in this case, harvesting takes place by licks made to one of 2 food tubes that are available. And we dispense food consecutively, trial after trial, for about 50 to 100 trials per food tube. To track the tongue, we use Deep Lab Cut. It's a fantastic Python toolbox that allows you to use convolutional neural networks to do markerless pose estimation. And with that, we have a representation of the video frame where we're tracking the nose, the tongue, the food, and the tubes. Here are some sample eye trajectories of saccades made to the variety of different primary and secondary targets. And of course, using Deep Lab Cut, we have a representation of the trajectory of the tongue, from which we can compute a variety of different kinematics in time, such as displacement, velocity, and angle of licks. Now, we noticed something rather fascinating, and once again, this came about rather serendipitously. That is a function, of course, of how we're giving these animals reward. And that's that, to get reward, it's actually quite challenging, and animals are oftentimes unsuccessful more than they are successful and require many, many licks to achieve satiety. So there's quite an effort cost associated with harvesting. in this case, much like we saw on the crows. Here's an example of a successful lick where the animal shoots its tongue out, aims it into the tube, and pulls a tiny bit of reward out. Now, I must note that these food tubes are positioned quite eccentric to the central gaze of the animal. And oftentimes, marmosets rather foveate by first making gigantic head movements as opposed to gigantic saccades. And of course, we train them to make saccades eventually. However, they very much more rely on sort of proprioceptively finding the tubes. And so oftentimes what we see is that they're not fixating on the tubes while they're making licks. They tend to use a scanning lick in the first few licks of a particular bout to encode the position of the tube in space in front of them. It's very, very fascinating. Here's an example of a few unsuccessful licks, again, which tend to take place more than successful licks. In this case, you see them overshooting a little bit. And in this case, you see them simply colliding with the tube and bouncing off and attempting a microcorrection, of course, unsuccessful. And because of this phenomena, because harvesting is quite effortful, we noticed a really interesting pattern that takes place in animals. And this sort of, we see them converge on a sort of strategy of doing this task that changes as they go from being naive to overtrained. Actually, just briefly, here's some success statistics. Both animals tend to be 30% successful in their licks, so it's fairly low. So as I mentioned, because reward was effortful to extract, we tend to see that subjects work for a few trials to accumulate food and then commence harvest. So they exhibit sort of delayed gratification, much like the crows deciding to leave small clams behind. Here's a sample of this in time. So x-axis is time, y-axis is food volume. Animals perform many successful trials to accumulate an amount of food in the tube that justifies initiating a licking bout, that justifies the harvest. They then initiate many, many licks to harvest that food, thus depleting the cache, and then repeat the cycle over and over again for about 1.5 hours to 2 hours. Here are some summary statistics of this. They typically attempt about 8 trials or so, both animals. We have subject M and subject R. These are 2 different marmosets. They typically are successful in about 5 trials, and they produce about 20 licks per bout. So it's a very rich data set here per session. Now, we then asked a question that was fairly an intuitive extension of this phenomena that we witnessed, and that's that, how does varying the effort cost of a harvest affect the decisions that the animal makes and the way in which they work? And we went about this by doing something fairly simple. We just basically made the food harder to get to by moving the distance of the tubes, okay? So in this case, and across this entire presentation, we're gonna have 8 different effort bins associated with the harvest. We have 8 millimeters, 10 millimeters, and 12 millimeters, and this is a quantity that describes the distance of the opening of the food tube to the mouth of the animal. 8 to 12 going from easy to hardest, of course. And first, we looked at the decisions that the animals made and asked, do they change how they do the task based on how hard it is to get to these food tubes? And what we noticed right away was that as effort cost increased, subjects sort of exaggerated this phenomena of working more, delaying the harvest, and then eventually harvesting when they thought that the food cache was large enough to justify the harvesting effort. So basically what we saw was that animals attempted more trials, and of course were successful in more trials, as we made it harder for the food to get to. Within these work periods, of course, there are 2 types of saccades made. There are the reward-relevant saccades that I showed you earlier, the sequence of 3 saccades. And then we have what are called other saccades, either just spontaneous saccades made while the animal's not making targeted saccades to the reward-relevant targets on the screen. The x-axis in this case is saccade amplitude, and the y-axis is saccade peak velocity. And we wanted to ask a question that was basically, are reward-relevant saccades different in any way in their vigor in comparison to other spontaneous saccades? And of course, what we noted was something fairly expected, which was that reward-relevant saccades in both animals across amplitude bins were more vigorous than spontaneous saccades. Now, within these reward-relevant saccades, how does vigor change as a function of the difficulty of the effort cost of that particular session? And we saw something rather interesting. So in this case, in the left column, we'll be looking at the vigor of the saccade and how that changes over time. X-axis is the trial number in a given work period. Remember, I mentioned that they attempt about 8 trials. However, in this case, I'm plotting for you 10. And we noticed in both animals a very, very interesting behavioral phenomena. That's that following a harvest, the first saccade of the work period tended to be high in vigor. And that vigor slowly decreased as the animal approached the next harvest. Further, these effects were scaled depending on the effort cost of that particular session. So when the session was easy, if you will, animals were more vigorous in making saccades than when it was harder. Now, this is really interesting. Why? Because the main effector affected here is the tongue, right? It's harder for the tongue to get to the tubes. However, we saw a change, a sort of global change in vigor in the eyes as well. And this was consistent across both animals. We then asked what's happening with the pupils and what might that tell us about the state of the animal as these trends in vigor are taking place. And so here I'm plotting for you again on the x-axis trial number during the work period, and I'm plotting for you on the y-axis pupil area, z-scored. It's computed as the average pupil area in a 500 millisecond bin around the trial. And what we saw was that the pupil size tended to change. The changes in pupil size closely track those of vigor, both in the decrease within a work interval, but also in a slight separation depending on the effort cost of the harvest of that given session. Basically, the animals were more attentive and aroused when the effort cost was lower during a task and when movements were more vigorous than when it was easier, than when it was harder, pardon me. We then asked what effect vigor had on saccade accuracy. In this case, accuracy represented as both magnitude of endpoint error as well as variance of endpoint error. And we saw something that fairly contradicts the speed accuracy trade-off argument, and that's that more vigorous saccades, that is saccades in which the pupils were actually more dilated, also tended to be far more accurate too. The error magnitudes and variance were smaller. They were both more accurate and precise with their saccades when the saccades were more vigorous. We're then going to take a look at how the animal harvests now, shifting away from the work period to see if some of these trends perhaps are similar in the licking system. So of course, as you can imagine, animals make two different types of licks throughout a task. There are reward-seeking licks, which is what I just showed you prior. These are licks made to the tubes with the intent of collecting food. And then there are other licks, grooming licks, sort of analogous to spontaneous saccades, if you will. These are movements not made to collect any reward and not really task relevant, but rather just to clean themselves because things get messy. And we asked a similar question, which was, how does this reward-seeking movement differ from grooming movements? And so on the x-axis for two animals in both protraction and retraction phases of a lick, I plot for you the peak velocity of each of these phases. And what we saw was that in protraction, across all amplitude bins, reward-seeking licks were more vigorous than grooming licks. Same applied for retraction phase. And generally speaking, the retraction of licks was more vigorous than the protraction. Now, this does also make intuitive sense because the protraction phase includes this very skilled component. And perhaps the animals have to be a little bit slower and more accurate in making the protraction phase of a lick, whereas retraction tends to be a straight shot back to the mouth. We're now going to take a look at reward-seeking licks and changes in vigor as the animal harvests. So we asked, how does varying effort cost of a harvest affect the way in which the animal harvests? What we noted was rather fascinating and sort of paralleled what we saw in the eyes. So in this column, I'll be plotting for you lick vigor as a function of lick number during a harvest. Recall that the animals make about 20 licks per harvest. I'm plotting for you extensions into 30. And we noticed something rather fascinating, the vigor patterns of the protraction phase of a lick. And that's that the early licks tend to be fairly slow. Recall, I told you that animals typically sort of make a scanning lick prior to continuously harvesting after that. And these initial licks tended to be fairly low in vigor. Once the animal encoded the position of the food tube, the vigor of the protraction phase of the lick in both animals spikes up and then slowly decreases as the animal harvests, depletes the food cache, and achieves satiety. These effects were further scaled by the effort cost of a harvest, as expected and as seen in the eyes. When the food tube was closer, the licks were more vigorous, and when the food tube was farther. Once again, like we saw in the eyes, the pupil size closely tracked these changes in vigor. So much so that the pupil size around the early licks were far smaller than around the later licks. And also what we did notice was that the animals were far more attentive and aroused when harvesting than when working. The z-scores here are all positive, indicating an increase from the mean of the session when referring to pupil size. We then asked what potential effect expectation of reward had on lick vigor. Now, what do I mean by expectation of reward? Well, the animals completing trials, some number of trials per work interval that precedes a harvest. And what you can see here are bins of trials completed in the preceding work interval. We have 2, 4, 6, 8, and 10. 10 indicating many, many trials being done, and thus more reward being accumulated. On the y-axis, I plot for you the vigor of the protraction phase averaged for the entire harvest. And what we saw again was that as the animals performed more trials successfully in a preceding work interval, thus expecting more reward, the vigor of the licks in the harvest to follow were higher when they expected larger reward than when they performed fewer trials and expected fewer rewards. And once again, that effect was scaled by the effort cost of a harvest. When the sessions were easier, the effective expectation of this reward through lick vigor was higher than when the sessions were harder. Finally, we asked, how does success history play a role in modifying the movements of the next lick? And I'll explain what I mean by that. So as I described to you guys, some licks were successful, like this. The animal made it into the tube. And some licks were unsuccessful, like this. For whatever reason, they didn't make it into the tube. And we're going to take a look at what success history, so each one of these outcomes in a previous lick, how that affects the next lick. Okay, so on this left column over here, we'll be focusing on lick vigor first. On the x-axis, the leftmost point is the previous lick, and the rightmost point is the next lick. And we have this plotted for both animals. In the left panel of the left column, we have successful previous licks. On the right column, we have failed previous licks. And what we saw was that the change in vigor for both animals following a successful lick was fairly large and positive. Whereas when the animal failed in a previous lick, the vigor either didn't change or actually decreased, depending on subject. There was some inter-subject variability there. We then asked what effect this might have on the pupils, what's happening in the eyes and in the state of the animal as they either succeed or fail. Interestingly, actually, is that when they succeeded, there were large sort of dilations in the pupil that closely matched the changes in vigor. However, when the animal failed, we did not see a constriction of the pupils or no change. We actually saw a fairly significant increase in pupil size. And this is fairly curious because it tends to be the first sort of contrasting result that we've seen here when examining this lick between lick vigor, this pattern, excuse me, between lick vigor and pupil size. And so I asked why this may be, and I delved into this to a fairly shallow extent thus far. And here's what I think is taking place. So, of course, not all licks are the same, and not all movements tend to be the same in terms of difficulty. And difficulty tends to put a different sort of strain in terms of cognitive load on the subject, right? For difficult movements, you would have to focus a little bit more. And of course, states of attention and focus and cognitive load are all variables that tend to affect pupil size as well. So there's two types of successful licks and failed licks, really. You can have an inner tube lick, excuse me, like this, that's done when the food tubes are fairly depleted, and when the animal has to aim its tongue into the tube to pull food out. And then you have outer tube licks like this, in this case, a successful outer tube lick, which takes place when the food has accumulated after a large flurry of successful trials, where the animal doesn't actually have to go into the tube, but rather they can lick towards the outside of the tube. Now you can imagine there are two very different accuracy demands on these licks, and thus these two licks are very different in their difficulty level, right? And what I found was fairly interesting and consistent across both animals. On the left panel, we have subject M. On the right panel, we have subject R. And if I was to plot for you the z-score as a function of effort cost of harvest, for both of these types of licks, you see that actually the pupils are quite a bit more dilated during the inner tube licks, the more difficult licks, than the outer tube licks. And I don't have a figure for it in this PowerPoint as of yet, but following a failed lick, the probability of inner tube licks and thus successes tends to increase as well. So that's what I think explains this phenomena that we see here. So then we asked how pupil size relates to movements across all conditions, across all effort conditions, across all movement modalities, and across all effort conditions as well, reward conditions, excuse me. What we found was that regardless of the period of the task, the movement modality, an increase in pupil size was a fairly robust predictor of movement vigor. So in the left panel, we have saccade vigor. On the right panel, we have lick protraction vigor. X-axes in both cases are pupil size, either during the work period or during the harvest period. Y-axes are vigor. And what we see were strong correlations between pupil size and vigor. Further, we asked how pupil size might be related to the decisions the animal made to either work or harvest. And what we found was very interesting. It was that pupil dilation corresponded to shorter work durations, but longer harvest durations. So when the animals were more attentive and aroused, they actually elected to work less and harvest longer. So in effect, sort of a link between both decisions and vigor of movements as related to pupil size and perhaps state of the animal at that time. So in summary, to conclude this talk, we explored the link between control of decisions and control of movements. We designed a foraging task in which marmosets worked to make saccades, accumulated food, and then harvested their cache via a licking bout, a foraging task, if you will. We then manipulated effort cost of a harvest and noticed a global change in the state of the brain. When we increased the effort cost, the pupils constricted, the decisions exhibited delayed gratification, and the movements displayed reduced vigor. So what? Where might this work lead us? So of course, there's a link between how we value options in a decision-making scenario, and the decisions reflect more than just how we value things, but we see sort of a difference in the way in which we move towards the option that we value more. And I must apologize in advance. This is a fairly text-heavy slide, so I won't be offended if your pupils constrict and your vigor completely tanks. But what we know is that both the choice that we make and the action that reflects that choice are part of a global cost that affects our fitness, theoretically speaking. In this case, the global cost I described was utility of a choice and action. Utility is composed of reward and effort variables, and these are both widely encoded in many regions of the brain, including brainstem neuromodulators such as dopamine, serotonin, and locus coeruleus norepinephrine. Now, when we look at vigor, we tend to have a very dopamine-heavy perspective on things, mainly because of the way in which dopamine encodes aspects of reward, such as reward prediction error, and there are tremendous links between reward prediction errors and effect on vigor of movements. However, perhaps seeing this link between movement vigor and sort of pupil dilation, we introduced the locus coeruleus norepinephrine system into the mix. Importantly, this system strongly correlates with pupil size and provides us with a fascinating, non-invasive proxy to measure internal state. Thus, we designed this experiment that allowed us to vary an economic quantity and then measure how the brain responds to these changes by altering its decisions, movements, and pupil size. So it's a task that allows you to get at all three of these things. Now, there's a question that we're left with, which is what might be the neural system that links decision-making circuits with those that control movements? And of course, we tend to have a dopamine-heavy view on things, at least thus far in the literature. However, in this work, we've seen that there are parallels in changes in vigor and changes in pupil size. And we know because control of the pupil is actually causally dependent on the activity of neurons in the brainstem neuromodulatory circuits, primarily the locus coeruleus. Our results raise the possibility that increased activity in these circuits that produce dilations in pupil impacts both the brain regions that control decisions, discouraging delayed gratification, and those that control movements, encouraging increased vigor. This is not to mention that the locus coeruleus, which is again tends to drive dilation of the pupils, also projects vastly to the cortex. But perhaps that's some stuff to get into for another day. So that's the end of my talk. It was a short and hopefully sweet one. I'd like to acknowledge my lab mates who are fantastic. Science is, of course, a team sport. Incu and Vivian helped us develop our deep lab cut pipeline to track the tongue, and we developed software together to do curation of the tracking of the tongue. Amin played a significant role in allowing us to analyze pupil data and develop a sort of pipeline for that. And of course, the senior lab mates, Ehsan, Jay, and Simon, not just for making giant contributions to our setup, but also for allowing me to pull from some of their behavioral data to add to mine. And of course, the captain of our ship, the fearless leader, Reza, for all that you do. And finally, our funding sources, because all of this is very expensive and requires money. So thank you very much, guys, and I'm happy to answer any questions that you might have.
Any questions for Paul?
Yeah, hi, Violetta. Please feel free to.
Yeah, hi. Thank you for the nice talk. I didn't know about this task, but it's really nice. I'm working with the skill reaching task, and I was kind of comparing. I'm working with rats, but this tongue reaching, if I might say, it's really cool. And what your work is also very nice. So I was just wondering methodologically if you also have a preference in the site they go to. I mean, is it an individual preference? Because in the skill reaching, for example, when they approach for pellets, usually the animals in the beginning develop a preference for one pole that they go, for example, left or right, and they keep going like this for the whole task. But I was wondering if in this kind of task, it's also like a tongue direction preference for the animals.
That's a really interesting question, and that's a question I get almost every single time. Yeah, so sort of handedness of the side of the tongues. I didn't notice anything sort of holistically, but what I did notice was, so prior to creating this setup where we had the two food tubes, left and right tubes, eccentric to the animal, we used to have just one food tube because we weren't quite interested in tongue movements. We were studying the eyes and the neurophysiology that drives eye movements. And so because of that, the animals sort of overtrained themselves on making rightward licks. And so when we then, in these overtrained animals that were making rightward licks, when we introduced a second food tube, we absolutely saw a preference, not just a decision-based preference to go to the right, but also the accuracy of the movements going towards the right. We noticed that rightward harvests tended to have more inner tube licks and licks that were successfully made to the inside of the tube than the left. It would be very fascinating, and we're actually doing this right now, looking into a naive animal that from off the bat is trained on both sides. And perhaps there is a handedness to the way that they lick. Interestingly, also, while these animals are doing the task, I'm also recording from their cerebellum, from neurons that care about licking. And we do see directional preference in Purkinje cell neurons, but also deep cerebellar nuclei neurons in the vestigial nucleus that modulate with tongue movements that is directional based. So who knows how that manifests. Maybe in these data we're collecting in these naive animals, we might see something interesting related to handedness.
Thank you.
You're welcome.
Paul, can I ask?
Yeah,
Great
Solomon,
talk
please go ahead.
Yeah, great talk as always. So you show when you show the vigor in the sense of the speed of the saccade as a function of the effort. Did you look at reaction time as well as a measure of this vigor?
Yeah, absolutely. So reaction time and vigor tend to go hand in hand. So much of the effects that we saw in the vigor, we also saw in the reaction time. Granted, the effects were a little smaller. And if I was to plot for you in all these saccades being made, vigor is, if I was to sort of plot reaction time versus vigor, you would see strong correlation. More vigorous saccades tend to have far shorter reaction times than larger, as is expected. Yeah, reaction time is a great proxy for vigor as well.
Cool, thanks.
May I? Sorry, I can follow up too.
Go ahead, Yolanda.
Okay, so I'm also interested in dopaminergic modulation in cerebellum, and I was just curious because you mentioned fastigial nucleus. Do you have like one hypothesis for which areas from cerebellum would modulate these vigor effects, or?
Yeah, it's hard to say it's hard to say which would modulate the vigor effect. So I've recorded from two regions. I've recorded from lobules 6 in the cerebellar cortex. This is a region that has, neurons, so Purkinje neurons, MLIs, GLIs that respond to licking movements. So namely lobule 6, a bit anterior to the oculomotor vermis. And we actually found that serendipitously. We were looking for eye neurons. And then in the deep cerebellar nucleus, a little bit anterior to the caudal fastigial nucleus, where the eye regions are, also we have neurons that respond to tongue movements. And I have yet to look deeply into the ephys of all of this, but that's hopefully the next project to come. We just wanted to kind of look at this behavior fairly closely first, but we have the data set ready to be looked at. Yeah.
That's great. Looking forward for the paper.
Thank you. Maybe hopefully next year I can have some more insight for you.
Aditya, go ahead.
Hi, Paul. I really loved your talk. I've been in communication with Professor Shadmehr, so I'm so grateful to have to attend this. I just had a small question. So when you were calculating the peak of the vigor of the saccades, the visual saccades, I understand that you measured that as the velocity of the actual peak velocity to the expected peak velocity for that amplitude bin. I was just wondering how you arrived at that expected peak velocity for each of these amplitudes that were measured.
Yeah, great question. So basically, what is vigor, right? So vigor is amplitude normalized velocity. When we say expected velocity, what you can think of is, okay, if we got all 5-degree saccades, looked at the average velocity of all 5-degree saccades, what is the ratio of the observed velocity divided by what we would expect from a saccade made in that amplitude bin? And why do we do that? Well, it's because amplitude confounds velocity, right? Larger movements tend to have higher velocities. There's a sort of hyperbolic relationship between these two kinematic variables. So to control for the effects of amplitude on velocity, we normalize it in that way. I hope that answers your question, Aditya. Thank you for it and for joining.
That really does. Yeah, thank you so much.
Josh,
Yeah,
go
Joshua, please feel free. Thanks.
Great talk. Really enjoyed that. It was wonderful. And thanks for having me as well. I had a question for what you found is that with greater distances or with shorter distances, you had more vigor. I was wondering if you looked also at accuracy for the different distances, because I was thinking about, kind of the Fitts law and speed accuracy trade-off. And as well, kind of related as if you're, if they're having to go further, it's maybe a slightly more complex movement and more planning. So how does that relate to your findings?
Yeah, that's a really good question. Yeah, so lick accuracy, I mean, there's a few different ways that we can go about measuring it. So one way is, of course, just outcome, if the tongue made it into the tube or not. And of course, what I did find, which I didn't present here and I'm still working on, is that when the tubes were further, they had lower probabilities of making it into the tube than when it was closer. So that's the first thing. I then also looked at accuracy measures, so like magnitude of the error of the endpoint of the tongue to the food. And I did find, of course, outcomes that paralleled what I just said about the outcome itself of the lick, which was that the error magnitude of a lick tended to be larger when the tube was further because they didn't make it into the tube and thus far the tongue was outside of it. We saw similar things in the eye actually as well, that in sessions, surprisingly, where harvesting was more difficult, the eyes were also a little sloppier. So the endpoint errors of the eye movements were actually greater in these sessions as well. I hope that gets at some of your questions. Please, by all means, let me know if it doesn't, and we can have a dialogue.
No, that does. Thank you.
Great. Thank you so much.
Well, thank you so much, everybody. I hope you have a wonderful holidays, beautiful Christmas. I hope you're safe and survive the next phase of COVID. And I hope to see you in about 2 weeks when we will resume the meetings. Thank you so much. Bye.
Merry Christmas.